Hi everyone. This is the fourth lecture for your chapter Respiration. In the last class, we saw fates of pyruvate. Okay. In fate of pyruvate, we discussed the anaerobic portion first, which included alcohol fermentation, lactic acid fermentation, and the second portion was aerobic respiration, in which we saw the first part of aerobic respiration, which is the Krebs cycle. I hope you have understood all of it, and with the help of that mnemonic, okay, the easier way to remember the whole pathway, the whole cycle was shown to you, okay, in the end of the lecture. Today we begin with the next part of this aerobic respiration, which occurs on the inner membrane of mitochondria, which is nothing but the electron transport chain. So why is that required? Before that, let us answer this question. We saw that at first glycolysis, so net product from glycolysis, we were getting 2 ATP. During the whole cycle, okay, whole Krebs cycle, you got only 1 ATP. So after glycolysis and TCA, we still have only 4 ATP in total. Okay, 2 ATP from glycolysis and 2 ATP from the 2 pyruvate molecules undergoing Krebs cycle. So we were promised a lot of energy, right? Where is all that energy? So how to generate more of this ATP? So that we will study in this ETC or ETS, electron transport system, and oxidative phosphorylation. The electron transport system, okay, it is a metabolic pathway in which electrons are passed from one carrier to another. Here, just like the electron transport system that we saw in case of photosynthesis, the same kind of transport system is present here as well. Okay, in case of the photosynthesis, the electron transport chain was present on the thylakoid membrane. Here, it is present on the mitochondrial inner membrane. Okay, right. So in this, the electrons, they are transferred from one electron transporter to the other electron carrier or transporter. Yeah, and this whole pathway, this whole path through which electron has to pass is known as electron transport system. The last electron acceptor, now there should be somebody who will accept the electrons in the end. So the last electron acceptor here is oxygen. Okay, so if you were wondering that where is the oxygen in the whole path, okay, remember the oxidation of glucose, the formula, it was C6H12O6 plus 6 molecules of oxygen giving you 6 carbon dioxide plus 6 water molecules, right? So where was this oxygen used? Till Krebs cycle it is used nowhere. Only in electron transport system it is used in the end, okay, as the terminal electron acceptor. It will accept the electron for well and good, okay, for once and for all. After accepting the electrons, it will get joined with 2 H plus, so 2 protons to make one molecule of water. Yeah. Where is the location of ETS? The location is in our mitochondrial membrane. And what is the role of ETS? The role of ETS is to oxidize and utilize the energy which is stored in NADH and FADH2. Okay, so the NADH and FADH2 that we produced from glycolysis, from link reaction, and from Krebs cycle, all of this reducing power, which is NADH and FADH2, will be oxidized here. And by their oxidation only, we are able to generate energy to drive phosphorylation. Let us study all of this one by one. Starting with the electron transport system. Okay, electron transport system is made up of 4 protein complexes. See, what does it mean by protein complexes? So this is not a single protein. There can be more than 50 proteins which are forming this one structure. Okay, so it is a protein complex, more than 2 subunits. Okay, more than 4 subunits are also present in the complexes. Now what is the function of these complexes? They pass the electrons, okay, E minus, and they transport H plus. They pass the electrons, so it is like passing the parcel. You take electron from one side, you pass it to the next one. So you pass the electrons and you transport H plus during the whole process. We shall see all of these 4 protein complexes one by one. Complex number 1 is known as NADH dehydrogenase complex. Okay, it is NADH dehydrogenase complex. Now stay with me. Okay, complex 1 NADH dehydrogenase, it looks like this. Okay, it looks like a gun, or it looks L shaped. As you can see, this is the inner membrane, mitochondrial inner membrane. On the lower side is your mitochondrial matrix. On the upper side is the intermembrane space, which is the space between two mitochondrial membranes. Clear till here? Now this L-shaped structure is shown in this green or blue color, turquoise color. Fine. In that you are also able to see these yellow and pink colored structures. This yellow and pink colored structures are nothing but iron sulfur clusters. Okay, so they are structures, or they are compounds made with iron and sulfur. So they are called iron sulfur centers or iron sulfur clusters. So the whole protein, okay, along with protein, it has these iron sulfur clusters. It has one molecule of FMN, flavin mononucleotide, FMN. What is the function of this FMN? It is a coenzyme, okay, or rather it is a prosthetic group. Prosthetic group meaning it is always found attached to the enzyme. So you have FMN, flavin mononucleotide. You have iron sulfur clusters or iron sulfur proteins. Fine. And then along with that you have other protein subunits. Now let us come to the reaction, okay, the reaction part or the electron transport part. So in the matrix, in the mitochondrial matrix, you have NADH from link reaction and from Krebs cycle. This NADH gets oxidized, okay, so dehydrogenase means what? You are removing hydrogen. So NADH dehydrogenase, this is that complex. The whole thing is known as NADH dehydrogenase complex. It will remove electrons and it will remove protons from NADH. The proton will be released in the matrix itself, okay, and only the electrons are transported. The proton is released free into the matrix. So NADH will get converted into NAD plus and the electrons are transported to FMN. From this FMN, the electrons are passed through the iron sulfur clusters and finally they reach something known as ubiquinone, which is this Q. Remember plastoquinone? So plastoquinone and ubiquinone are similar type of molecules. Okay, so here is your Q. Q is nothing but ubiquinone. Now when these electrons are being passed, okay, when the electrons are being passed through the iron sulfur centers, this NADH dehydrogenase complex, it opens a channel. Okay, there is opening of a channel, and when that channel will open, 4 protons, so 4 H plus, they are moved outside from matrix to inner membrane space. Okay, intermembrane space. Clear till here? NADH, the electrons are moved from NADH to FMN, FMN to iron sulfur clusters, and finally to ubiquinone. During this process, H plus, which is released in the matrix, 4 H plus, 4 protons, they are transported through this NADH dehydrogenase complex from the matrix to intermembrane space. Now, when ubiquinone, it accepts an electron, it also accepts 2 protons. So can you see this 2 H plus? So these 2 H plus the electrons coming from iron sulfur cluster and the ubiquinone will form QH2, which is reduced ubiquinone or ubiquinol. Fine. Clear till here? So NADH dehydrogenase complex. The second complex is succinate dehydrogenase complex. Now remember, I had told you in last class that this is a special enzyme. It is the only Krebs cycle and electron transport chain enzyme which is common. Okay, it is common between Krebs cycle and electron transport chain. Second thing, it produces FADH2, and third thing, it is found on the inner membrane of mitochondria and not in the matrix. Chalo, let us find out how the structure looks like. So this blue colored part, okay, is the protein forming the succinate dehydrogenase complex. As you can see, it has an FAD, okay, which is a prosthetic group of this enzyme. So this FAD is always found attached to the enzyme. Along with that, it has iron sulfur clusters or iron sulfur centers. And finally, it has a heme group. Heme group is nothing. Heme means what you find in your hemoglobin, okay, or you find in the cytochrome. The same kind of heme group is found here. Chalo. Now let us see how the transfer is taking place. So from succinate to fumarate, remember it is a reaction from your Krebs cycle. So when succinate gets converted into fumarate, FAD gets converted into FADH2. So FAD is reduced and succinate is oxidized. This FAD plus, it donates its electrons to the iron sulfur cluster, and as soon as it gives its electrons, it turns back into FAD. Clear? Now this electrons will pass from these iron sulfur centers back to ubiquinone. Okay, so it is again going to Q, and with help of Q and electrons plus 2H plus, it will give you UH2 or ubiquinol. Clear? So this is the same between both a complex 1, which is also producing QH2 in the end, which is reduced ubiquinone, and complex 2 is also producing QH2, which is reduced ubiquinone. Clear? Complex 2 does not transport any proton into the intermembrane space. So no protons transported in complex number 2. Right, so this is the reaction ubiquinone. So Q plus electron plus proton gives you ubiquinol or QH2. Ubiquinol, it gets oxidized to ubiquinone. Okay, so again the protons H plus are removed and the electron is transferred to the next complex, which is cytochrome Bc1. So complex number 3 is cytochrome Bc1 complex as shown in this diagram. This cytochrome Bc1 complex, if you remember, I had told you cytochrome is nothing but it is a protein which has heme group or porphyrin, right? Now heme group can be of multiple types. It can be A, B, C, D, E. So here the heme groups that are found are of two types, B and C1. If you remember in photosynthesis, we had seen cytochrome BCF, okay, B6F. So there were two cytochromes, cytochrome B6 and cytochrome F. Here you have cytochrome B and cytochrome C1. So together they are known as cytochrome Bc1 complex. It looks like this, okay? It has these two parts which are same as each other on the left and on the right. So here you can see these are the heme groups, okay? You have cytochrome B and cytochrome C1. Fine. So apart from that, it also has an iron sulfur cluster, okay? So FeS is shown here. So two cytochromes, cytochrome B, cytochrome C1, and two iron sulfur cluster. Now what is the reaction that is happening? Remember, during the complex 1 and complex 2, we produced reduced ubiquinone or QH2. This QH2 will come here, get attached to cytochrome Bc1 complex, and it will transfer its electron to the cytochrome Bc1 complex. As soon as it transfers its electrons to cytochrome Bc1, it again gets oxidized back into ubiquinone. Clear? So this cytochrome Bc1 complex, okay, complex number 3, during this process, it will now have the electrons. These electrons will be passed on to the next protein, which is called cytochrome C. Now when it passes this electrons to cytochrome C, which is shown in this dotted line here, when it passes the electron to cytochrome C, during this time it also transports 4 proton from matrix to the intermembrane space. Yeah. So remember this, apart from Nadh dehydrogenase complex, cytochrome Bc1 complex also transports 4 protons, okay, from the matrix to intermembrane space. Now the fourth complex, okay, till now what have we seen? We saw that complex 3 gave its electrons to cytochrome C. The fourth complex is called Cytochrome C Oxidase. Okay, So what does this cytochrome C oxidase look like? It looks like this. Okay, it has these iron or heme groups. It has copper also. So this is something new for you. Okay, it is a copper containing enzyme complex. Clear? What will happen here? So remember there are the cytochrome C which was present here. Okay. So complex 3 gave its electrons to cytochrome C. This cytochrome C is a mobile protein. Okay, it can move around between two different proteins. So this cytochrome C is reduced form. Okay, so it has electrons. These electrons will be passed to the copper of your complex number 4, which is cytochrome C oxidase. This cytochrome C oxidase has one cytochrome A. It has two copper centers. So here Cu, and one cytochrome A3. Here the cytochrome C gets oxidized, so it will pass its electron and become oxidized, and the electrons are finally transported through all of these copper and iron centers or cytochromes to the oxygen. So here you have oxygen O2 plus 4 protons on the matrix side, so 4H plus oxygen, and the electrons that you get from the cytochrome C oxidase or complex 4 will finally give you 2 molecules of water. Here also during this whole process 4H plus are transported out of the membrane, okay, from matrix to intermembrane space. H2O is released in the matrix, and this is the whole reaction. 4H plus 4 electrons, okay, shown here 4 electrons give you plus oxygen give you 2 molecules of water. As you can see, this is the free energy diagram. Okay, so on the y-axis you have free energy. Okay, so it is just showing you the energy, okay, the energy level, and on the x-axis you have energy flow. If you remember, I had shown you reduction potentials in photosynthesis, right? The same thing is shown here. NADH has the highest energy. From NADH, it is passing its electron to FMN. FMN passes it to ubiquinone. Ubiquinone passes it to the cytochrome Bc1 complex. Then from Bc1 complex, cytochrome C. Cytochrome C to cytochrome A and A3 of cytochrome C oxidase complex. Okay, and from that cytochrome C oxidase complex, it is finally given to the oxygen. Clear? So you can see there is some reduction in the energy. Okay, there is some reduction like this. This makes sure that electrons are only passed from one direction, that is from NADH to oxygen. Okay, it cannot go in this direction. From oxygen you can never take electrons and give it to NADH. It will only flow in this one direction. Clear? Fine. So this is the diagram that is given in your textbook. Let us very briefly go through it also, because there can be questions which can be asked from this diagram also. Clear? It is a quick revision. So if you want a break, just pause, take a deep breath, and then let us continue. Here on the left hand side is your intermembrane space, okay, which is the space between the two membranes. So this here is the intermembrane space. Right. Then you have the inner mitochondrial membrane and the mitochondrial matrix on the right hand side. On this mitochondrial membrane, which is shown in blue color, you have these 4 complexes, 1, 3, 2 and 4. We will see why this sort of arrangement is shown. Okay, now let us understand what will happen. Let us say your reducing power is NADH. Okay, we are talking about NADH. So you have this NADH. You want to oxidize this NADH. So which is the first complex that you go to? You go to the complex number 1, which is Nadh dehydrogenase. What will Nadh dehydrogenase do? It will remove the proton, so H plus will be removed, and it will transfer the 2 electrons. Okay, it has 2 electrons, so these 2 electrons will be transferred to FMN. Remember this FMN? This FMN will transfer the electrons to iron sulfur cluster, And iron sulfur cluster will give the electrons to ubiquinone, which is written here as UQ. Okay, fine. Okay. Fine. So we just saw from NADH electrons to FMN, FMN to iron sulfur to ubiquinone. When the ubiquinone gets electrons, it gets converted into UQH2, which is reduced ubiquinone. Fine. Now this reduced ubiquinone, it will give its electron to complex 3. Okay. We are talking about NADH oxidation. So from complex 1, the electrons will be passed through ubiquinone to complex number 3. Ubiquinone is a lipid soluble molecule, so it can move around. Okay, it can shuttle from complex 1 to complex 3. It will move around. It will take electron from here and then it will come here and give the electron. Fine, pass the electron. Okay, complex 3 is cytochrome Bc1 complex. So what will happen? The electrons from the ubiquinone, they will be passed to cytochrome B. Okay, here, from here to cytochrome B. Cytochrome B to iron sulfur cluster to cytochrome C1. Now this cytochrome C1 will give the electrons to cytochrome C, which is another protein found on the intermembrane space side. Okay, it is found on the intermembrane space side. During this process, there are 4 protons which are pumped out. Okay, so 4 protons from matrix, they will be pumped out into the intermembrane space. Now this cytochrome C again is a movable protein. Okay, it is a peripheral protein. Okay, you remember the 2 types of proteins that we have studied in cell membrane. Okay, so it is a peripheral protein, and this cytochrome C with the electrons, it will go to the complex number 4. So from 1 we went to complex 3, and complex 3 to complex 4. Complex 4 is cytochrome C oxidase. The name only suggests what it will do. It will oxidize the cytochrome C, which is shown here. This cytochrome C, as soon as it gives the electrons, it donates the electrons, it becomes oxidized. Clear? So cytochrome C will become oxidized. It will give its electron to copper. From copper, it will go to cytochrome A, cytochrome A3, and copper B. Now this copper B will transfer its electron to oxygen. Okay, so it will give its electron to oxygen. Oxygen with the electron and proton will now form H2O. So if somebody asks you, where is the terminal or who is the terminal electron acceptor? It is oxygen. Where is this happening? It is happening at complex number 4 or cytochrome C oxidase complex. Clear? One thing to remember is during NADH, okay, if you are talking about NADH oxidation, complex number 2 will be omitted. Okay, it is not required. From 1 to 3 and 3 to directly 4. All of these 3 complexes, they are pumping out 4 protons and 2 protons respectively. Clear? So these many protons from the matrix are moving into the intermembrane space. Now let us see what will happen if you have FADH2. Okay, let us say you have FADH2. So this FADH2 will go to your complex number 2, which is Succinate dehydrogenase complex. So succinate gets converted into fumarate, and during this process the electrons are transferred to FAD. Okay, from FAD they are transferred to iron sulfur cluster, and from iron sulfur cluster they go to the ubiquinone. Fine. So again, same as NADH, which was giving its electrons to ubiquinone, okay, through NADH dehydrogenase. Here also that is happening. So ubiquinone becomes reduced ubiquinone, UQH2. This ubiquinone will give its electrons to cytochrome Bc1 complex, which is complex number 3. So from 2 you have complex 3, and from complex 3 it will go to complex number 4. Okay, how? Let us see. So ubiquinone will transfer electrons from this succinate dehydrogenase to the cytochrome 3 or cytochrome Bc1 complex. Same thing will happen, cytochrome B, then iron sulfur cluster, then cytochrome A1, and then it will give its electrons to cytochrome C. During this process, 4 protons are pumped out. Now this cytochrome C will shuttle, it will go to complex number 4. It will give its electrons to copper A, from copper A to cytochrome A, then cytochrome A3 and copper B. Copper B will finally give its electrons to oxygen, okay, and form H2O. This complex also, it is releasing 2 protons from matrix to intermembrane space. So this is the reaction, or these are the steps that will be taking place if you have FAD as reducing power. Clear till here? So just focus on this diagram. This is the whole process, okay, shown in very quick form. Okay, let us start from here. You have NADH. NADH is giving, okay, just a second. Right. NADH will give its electron to NADH dehydrogenase. Protons are transferred from NADH dehydrogenase to ubiquinone, ubiquinone to the complex 3 to cytochrome C, cytochrome C to cytochrome C oxidase, and finally they will be given to oxygen. So that is the whole process that is taking place in the in this video. Okay, it is a little fast, but you just let us see it once more. Clear? So now we shall discuss the types of types of phosphorylation. Now we saw electron transport chain as well, but there also there was no ATP which was generated. So where is this ATP going to get generated? Let us understand. So there are 3 types of phosphorylation in biology. The first one is called substrate level phosphorylation. Okay, substrate level phosphorylation is when an enzyme like this pyruvate kinase enzyme, okay, of glycolysis, the substrate is PEP, phosphoenol pyruvate. From the substrate itself, okay, this phosphate group which is attached to the substrate, this phosphate is given to ADP. Okay, then it is called substrate level phosphorylation. From the substrate, phosphate is transferred to ADP and this gives you ATP. Substrate level phosphorylation, we have seen 2 reactions in case of glycolysis, from 1,3-bisphosphoglycerate to 3-phosphoglycerate, and second reaction was this one, phosphoenol pyruvate to pyruvate. So these 2 reactions produce substrate level phosphorylated ATP. Then in case of Krebs cycle, succinyl CoA to succinate, okay, so succinate synthase enzyme, it can also produce 1 GTP which is equivalent to 1 ATP. So these are all substrate level phosphorylation. Then comes photophosphorylation. Photophosphorylation we have already seen in case of photosynthesis. Okay, in presence of sunlight, phosphorylation will take place, so it is known as photophosphorylation. And the third and the final type, which is important for respiration, is called oxidative phosphorylation. Why is it called oxidative? Because you require oxygen, okay, as terminal acceptor, as we just saw. So oxidative phosphorylation. Let us study this thing. Okay. Before studying, this is just a small comparison between the electron transport in chloroplast and electron transport in mitochondria. If you remember, in case of chloroplast or in case of thylakoid, from PS2, which is photosystem 2, 2 electrons were getting donated from H2O. So H2O was breaking down into oxygen plus protons, and the electrons were transferred to photosystem 2. From photosystem 2, they were passed on to plastoquinone to cytochrome BF, from cytochrome BF or B6F to plastocyanin. Plastocyanin will give electrons to photosystem 1, and photosystem 1 was finally giving it to the enzyme which is called NADP reductase, and the terminal electron acceptor was NADP plus. Clear? So this NADP plus will accept electrons and get converted into NADPH. Here we are actually doing the opposite, right? So the substrate here is NADH, NADH will donate its electron. They will be passed from the complex 1 to ubiquinone, which is Q. From ubiquinone, it will be passed to complex number 3, which is cytochrome, um, cytochrome Bc1 complex. Okay, so from cytochrome Bc1 complex, they are given to cytochrome C, which is same as plastocyanin. Okay, it is just called cytochrome C. Now this cytochrome C will pass its electron to the complex 4, which is cytochrome C oxidase. Complex 4 will pass the electron to oxygen, and this oxygen will get converted into water. So can you see, it is quite the opposite reaction of the same thing. Okay, photosynthesis, we studied the terminal electron acceptor was NADPH, and here the terminal electron acceptor is oxygen. Right. So chemiosmotic hypothesis and oxidative phosphorylation. Remember, we have studied chemiosmotic hypothesis in case of photosynthesis as well, right? So ATP synthesis is linked to the breakdown of proton gradient across a membrane. So now till here we have seen that this was the membrane, inner membrane. 3 complexes were pushing out the protons, okay, into the intermembrane space, because of which there is higher proton concentration outside in the intermembrane space and lower concentration in the matrix. Because of this, there is this sort of gradient, okay, and if you want to break the gradient through that process, okay, through breaking the gradient, you can get energy. We shall see that now. Okay, just think of it like here is a water tank, okay, and the water tank is filled and this portion is at lower level. So through this water tank, when the water will flow downwards, during this process, if we put a turbine here, then that turbine will produce more and more electricity, right? Same way, same process, just apply it here. Let us study. So H plus gradient across the mitochondrial inner membrane is generated by. So how is this H plus gradient generated? So 10 H plus or 10 protons are transported to the intermembrane space. This is the intermembrane space during electron transport from complex 1, 3 and 4. Complex 2 does not transfer any any protons, okay. 2H plus and half oxygen molecule is used to make water. So this is taking place in the matrix. So in the matrix, H plus is being utilized. If H plus is getting utilized to make oxygen and proton into water, so there is less proton on the inner side and there is more proton on the intermembrane side. So this gradient, there is a gradient. Now there are high proton molecules in the intermembrane space and lower proton molecules in the matrix. So this gradient is broken down by movement of the H plus, by movement of this proton into the matrix. Okay, they can move back towards their what you can say, downhill. Okay, downhill means from their high concentration to their low concentration, they will be able to move. But this movement is through FOF1 ATPase or F0 F1 ATPase. Clear? It is also known as complex 5 and it is the final complex in the electron transport chain and oxidative phosphorylation. When this H plus is transported through this FOF1 ATPase, ADP, okay, adenosine diphosphate plus inorganic phosphate is converted into ATP. So in case of mitochondria, okay, here the H plus gradient or H plus concentration is high in intermembrane space. In case of chloroplast, the thylakoid space, okay, or thylakoid lumen had higher H plus. Fine. So this is the whole process. During electron transport, you have pushed out the H plus. This H plus will try to come back in the matrix through this ATP synthase, and during the process, ATP will be synthesized. I don't know if this will work. This was supposed to be animated, but for some reason it is not working. Anyway, so this is the structure of F0 or FO. Okay, FO, as you can see, it is a rotor-like molecule. It is a rotor-like protein which is embedded in the mitochondrial inner membrane. Then you have this F1 particle which comes out like a lollipop. Okay, it is like a lollipop. Now, whatever this will be on the matrix side. Okay, the lollipop, this F1 particle is on the matrix side. This whole structure is known as oxysome, if you remember. Okay, so now proton, which is higher in the intermembrane space, it will get attached to the F0. F0 will rotate. Okay, it will go round and round. When F0 rotates, this F1 also rotates. And when F1 rotates, ADP is converted into ATP. After one full rotation, the H plus will be coming back into the matrix. Okay, so this H plus gradient, okay, this movement of H plus is used to turn this turbine, and with turning of this turbine, you are producing ATP. F0 is a membrane protein that forms channel for H plus to cross. Okay, so it is basically like a channel. F1 is it juts out meaning it comes out in the matrix and has site for synthesis of ATP. 4H plus, so 4 protons are required to pass through the ATP synthase per ATP. So if you want to synthesize one ATP, you should at least have 4H plus which should pass through the ATP synthase. So 4H plus will pass through this ATP synthase or complex 5 and give you one ATP. So that all the tally and how many ATP are synthesized, how many NADH are converted into ATP, all of that we shall see in the next class. Let's see you next time.